exercise 14 and 15, 14 is motility, 15 is oxygen requirements. You'll notice there's a water bath at your table. If you could just bump up that temperature there so we can get to the water boiling, that would assist us in this process.
I'll use my body
What I will
language
ask
to use
is
her
that,
words.
you guys come up and get your plates
How
from
about like
Monday.
135 or put it in like this thing?
I'll just bump it up. The lights to lights.
Like 430?
Yeah.
I'm always this kind of stuff. I can't be I can't be a
Get
person
your
who can
plates
and
back
stuff.
to your
Oh,
table.
virtually, no.
What I
can't.
want you to do, because right now I believe they're separated by organism, what I want you to do is separate them by media. So we should have 5 stacks of 4. Right now you probably have 4 stacks of 5. So I give you 4 stacks, right? We need one more?
Yes.
Yeah, that's all right. You probably have 4 stacks of 5. What I want you to do is separate those out into 5 stacks of 4 by media, okay? By type of media. Because we had 5 medias that we put together here. So group them by the media. TSA with 10% sodium chloride, MSA, MAC, and blood auger. Okay?
What is it blinking? Because it's getting hot.
Because you're hot. It's winking at you. That's how bored I am. I'm entertaining with cheesy jokes.
Separate them out by media. Just be careful because there is a TSA and a TSA with 10%. So make sure you don't mix those.
I think what we do was we got a new person and then
We're
one
going to
day
go
they
through
just
all
quit.
of these. We're going to talk about each of them.
I was just thinking about it. Like,
We are going to be recording our results on page 93 and 94. So we're going to go through each of these as we do so. Okay, we're going to start out with tryptase soy agar, the TSA.
okay, guys. You better, yeah.
So we're looking at TSA by itself. Now you'll notice when you're recording your results, we have observations and we have interpretations. Observations are what you observe, what you see, and interpretations are how we take that information and provide ourselves with information. Okay, so we're going to start with the TSA plate. This is a general media. Observation would be growth or no growth. What do you see for your plates? Growth for? I only see yellow. Huh?
I'm sorry, what page are we in?
At 93. Which organisms show growth? All of them.
Mm-hmm.
That is correct. They should all show growth for tryptase soy agar on the tryptase soy agar plate. And this media is a media that is not specific. It just has a mixture of nutrients that support all sorts of organisms. And all of these 4 that we did should grow on this media. Okay? Your interpretation for that Well, they grew, so it's a live culture. These are live cultures that are able to utilize what? The nutrients on tryptocase soy alder. Something along that lines. All right. The next one is the blood auger plate.
Wait, where are we putting on interpretation?
That they're live cultures that can utilize the nutrients in that general media. They can grow on TSA. All right. The next plates you're going to look at would be, so you put TSA off to the side, we're done with it. That's as far as we're going. The next set of plates are going to be the blood auger plates. Okay? So the blood auger plate, this is an enrichment media as well as a differential media. So you have two bits of information. One, does it grow or not grow? Right? And then the second bit of information for the observation is, what type of hemolysis do you see? So if you recall from the discussion on Monday, right? Hemolysis is the breakdown of red blood cells. And it is a beta hemolysis is where you're going to have complete breakdown of the red blood cells. You can hold the plate up to the light, you'll be able to see right through that plate, around where the organism is. Not where the organism is, because where the organism, it's not a translucent colony. So where the organism is where those cells are located. But those organisms then produce an enzyme or toxin that they disperse into the environment, and so the surrounding environment around that colony of the growth will be clear, right? The rest of the plate will show red for the red blood cells that are present, but there's a clearing. That's beta hemolysis, complete breakdown of red blood cells. You can hold it up to the light. I have held it up to, say, the edge of the table where you can see the edge of the table in the background, right? When you're taking a picture. Okay, but you can see through the plate. Alpha hemolysis is where we have partial or incomplete hemolysis, and it produces a cloudy zone that's kind of greenish looking around that colony. Now, FYI, don't look for the hemolysis pattern. Don't look at the area where there's thick growth. Because the shadowing of the growth itself is going to give a greenish appearance. What you want to do is look at colonies, isolated colonies. Do they have a hot halo, a green halo? If you have to open the plate to actually look, do that. Okay? Open up the plate to look at the colonies, the individual cells. Do you see a halo around cells?
No.
And then gamma hemolysis is no hemolysis at all. Okay? In this case, when you look at a colony of growth, as you move from the media towards the colony of growth, there will be no change in the color of that media.
Does anyone have this staff?
So, to start off with, for your observations, number one, enrichment media, which organisms grew on these plates? The enrichment media fact is growth or no growth. Which organisms grow on the plate? All of them, thank you. All of them grew on the plate. Now, as far as the hemolysis pattern, so this is the second bit of information that you get from this plate. The hemolysis pattern has to do with the differential component, the blood cells being present, the red blood cells being present that might be broken down by the bacteria.
I think the second one.
Which one showed beta hemolysis?
There's no growth. There's no.
Staphylococcus aureus.
That's what I put,
Absolutely.
yeah.
We're going to do a lab later in the term. I'll tell you, it's kind of sad. Scientists are so mean. There's a toxin Staphylococcus produces. It's called alpha toxin. and alpha toxin causes beta hemolysis. Man, they should just call it beta toxin, right? We make their life so much easier. But no, it doesn't go that way.
Oh, I don't have my
What's
gloves
the
on.
salmonella?
Which one do you see alpha hemolysis?
E. coli?
Enterico? Salmonella, enterico?
And then Aureus is beta. And then salmonella is alpha.
Salmonella epidermidis is gamma. Alright, salmonella I would put as an alpha also. So, the two alphas are Escherichia coli and Salmonella entericus. And then we had enterica, excuse me, and then Staphylococcus epidermidis, the plate I just saw, definitely looks a gamma, right? Everyone agree? Of course you did, because I just told you around the teacher, and you're like, yes, you're right. No, I'm not always right.
Wait, say that again?
Sometimes I am. You better agree.
Which would be Selma is I got my salmonella as an alpha, because it looks green as a pond. Could that be a mistake?
Salmonella enterica and Escherichia coli are alpha hemolysis. Staphylococcus epidermidis is gamma.
Gamma.
That's and then Staphylococcus aureus is beta.
Oh, I don't know.
So alpha, alpha, beta, gamma. Don't go in that
So in
order.
this case, interpretation is all four of these organisms can grow in the on this enrichment
And then the
media.
Aureus is beta.
And differential would be the fact that, you know, the observation is the green tone haloing, the clear haloing, no haloing, versus the interpretation is beta hemolysis, gamma hemolysis.
Oh, I'm writing that.
Right, alpha hemolysis. All right, the next one we're gonna look at is the salt auger. The salt auger is TSA with 10% sodium chloride. Remember what I told you last week? Last lab. 10% sodium chloride is what you'd have to give on a test if you were asked, right? Sodium chloride can be spelled out completely, or you can use the molecular formula. Don't care as long as the molecular formula is correct. But you can't tell me salt, right? If you say 10% salt, you're not gonna get that credit. Okay, because there's more than one type of salt. So you need to be specific. Now in this case, this is a selective media. The high level of salt is going to inhibit certain microorganisms. So what we're going to be looking at is, do these organisms grow or not grow? So which organisms grow on this media? This one. Correct. The two Staphylococci. The interpretation is they are halotolerant. E. coli and Salmonella enterica do not grow, and therefore they are not halotolerant. Okay, that's the interpretation. All right, so now we're going to the mannitol salt auger. Mannitol salt auger, that MSA plate, is a selective media and differential media. So again, there's two things you're looking for. The selective media component is this has 7.5% sodium chloride. So, once again, which organisms grew? The Staphylococci, and therefore the interpretation is that they're halotolerant. Ooh, big surprise. We've already determined that, right? From the other media. But this media also has a second component to it, and that's the differential media. We are looking for fermentation of mannitol. If you see yellow, right, that's that pH indicator, phenol red, turning from that red color to a yellow color because the environment has become acidic. So if you see yellow, that means the organism ferments mannitol. So the interpretation, the observation is yellow, the interpretation is it ferments mannitol. If you see no color change, so it's still pink in color, right, which is really no color change, then it does not ferment mannitol. Which organism ferments mannitol? Staphylococcus. And Staphylococcus epidermidis does not. Okay? So again, because anytime you have two components like that, selective media and differential media, there's two bits of information, right? Does it grow or does it not grow? And then the second is, does it then do that particular activity or does it not do that particular activity? In this case, ferment mannitol, yes. Your E. coli ferment, E. coli doesn't even grow on this plate. Should it? So there had to have been an
What
issue.
if it's just old?
Then contamination. Yeah, E.
Which
coli
one is that?
can't tolerate that high level of 7.5%.
This is epidermis.
No, she said no.
No, no,
It's not.
I am going to, let's go through all of them first, and I'm going to cover what Okay. All right. Now, the last set of plates is the McConkey auger plate. So the McConkey auger plate is selective, so it has two components, the selective aspect and the differential. The selective aspect is the fact that it has bile salts, right, and crystal violet, and those inhibit gram-positive organisms. Thereby, you're selecting the gram-negative. Which organisms grew on these plates?
Salmonella.
Correct. Salmonella and E. coli, which both happen to be gram-negative organisms. The next observation that you're doing is looking at the differential media component, and we're looking at fermentation of lactose. Okay, and we have the pH indicator neutral red in this media. Neutral red turns a powdery pink if the environment becomes acidic. So which one turned a powdery pink?
Powder pink. E.
E.
coli.
coli turns a powdery pink. The other one has no color change, or sometimes it actually looks a little like a yellowish color change, but that's not the reaction we're looking for. Okay, it does just due to the end products of its growth, right on that media. It does change it somewhat from the original color, but that is still not. We're looking for that powdery pink color.
So does it go as halo tolerant as well if they have?
Halo tolerant has nothing to do with this.
This is
There is no, we're not talking about sodium chloride here. We are talking about the selective component is the bile salts and the crystal violet that inhibit the gram-positive cells. So those that grow are gram-negative cells. So if you see growth, it's gram-negative cells. Okay, so there's a difference as to what's in the media to provide selective aspect as to what if the interpretation means, right? For salt auger and mannitol salt auger, growth due to the selective component meant it was halo tolerant. In this case, the two components that make it selective means that the organism is a gram-negative organism.
I don't know what you're telling me, but from underneath the lactose
Has nothing to do,
chapters,
you're looking for powdery pink.
this is
13,
Has nothing to, don't
I believe 13.
bring
14.
in mannitol salt auger, the yellow color that is produced by mannitol salt auger, as
Yeah,
part of
because we're gonna
this
do the
test.
14
This is a
today.
different test. It also is a different pH indicator. This is neutral red. Neutral red in acidic condition turns powdery pink.
What's the aureus?
Alright, so
The monkey.
the appearance that you're looking for an acid, that's the acidic environment is not yellow
McConkie.
in this case.
Which
It's a
organ
different pH indicator.
Aureus.
And the pH indicator, if it is an acidic environment, is
epidermis
that
as
powder
well.
green color.
Yep.
Alright, so, in regards to questions for this, I'm not going to ask you to, well, that I should. I'm not going to ask you about the organisms, so I'm not interested in you memorizing the organisms so much as the medias, what category do they fall under, right? Selective, differential, general, that type of thing, enrichment. What that means, right, to be selective. What does it mean to be differential? What does it mean to be a general purpose or an enrichment media? And then, in that media, if it is, say for instance, mannitol salt agar, if it is mannitol salt agar, what makes it selective? What makes it differential? And if you see a plate, can you interpret the results? Okay? Not, can you give me the organism names? I'm more interested in you being able to if you have a plate in front of you of mannitol salt agar and you're looking at it, can you tell me what that plate means? It's growing, so it's halotolerant. It's yellow, so it ferments mannitol. Okay? I could ask you, what makes it differential? Well, the presence of mannitol and the presence of phenol red, the pH indicator. That type of thing, okay? Not, what did salmonella do on this media? Okay, I'm not gonna ever ask that. Or am I gonna show you a plate and say, what organism is this? No, okay? I can show you a blood agar plate and say, what type of hemolysis pattern is this? You need to be able to differentiate the 3 hemolysis patterns. Okay, does that make sense? So don't get tripped up and locked up in organism names. It's not what this is about. It's the fact that we use different media. They are different. So the question comes up, well, this is kind of yellowish looking. Does that mean it's acidic? No, because it's a different, it's a different media compared to another media. This one has neutral red. Neutral red is a certain appearance in acidic condition, right? So that's what I'm looking for. Not, can you tell me this organism, what's it going to look like on this media? All right? Questions? All the plates will go into the biohazard bag. Okay? The only slide that I have left while you guys are doing that is just a compilation of what you guys saw. Okay? So you have that in your, or on Canvas for you to be able to view. That's all the PowerPoint that we have.
Can you make the last one?
This is on there.
Yeah.
This is what we're gonna get on next.
Oh, I can.
Yes, the quiz on, I'm pretty sure. There's 2 quizzes next week, on Monday and on,
Monday, it says only lab quiz.
Only lab quiz and then
I know, but there's 2 lab quizzes next week, Monday and Wednesday.
Yeah, because Wednesday we have lecture
So,
and lab.
pay attention as to what you're going to be on that Monday quiz as compared to Wednesday quizzes. Okay? That, you just look at the week and it tells you, what is actually going to be covered in this quiz. Now, so this here is actually provided to you. It's obviously the Staph aureus, Staph epidermidis, Salmonella, Escherichia coli for the different media. The only one I don't have a picture of would be the hemolysis patterns, but I have them here. You'll notice this one, you can actually see through the plate. So you see the countertop, and this is actually, I was probably standing right here while I was holding the plate, taking the picture, okay? So you can see through the plate, okay? As compared to this one, as you approach the individual colonies, you notice there's no haloing effect. And I know it's hard to see it as it expands, and I turn on the lights for a moment. Maybe that doesn't help. No, it doesn't help. But when you look at it online, you'll see there's a haloing effect. It may not be as green in this picture, but you can see a fuzzy haloing effect around those colonies. That's the alpha.
So one is what we're looking for, if we were looking for alpha, I think gamma will be number 3.
They're in order, alpha, beta, and gamma, in this figure. Okay? All right. Because everyone's water's going, if I can have somebody at each table get 3 of the screw cap tube, the fluid thioglycolate, put it into the water bath, it needs to boil for 5 minutes, and then we gotta take it out and let it cool. Okay?
You okay?
Yeah, my front.
So it's 5 minutes, right?
Yeah.
Yeah, just put it in, if it goes a little more than 5 minutes, it's not gonna be an issue, okay? Like I said, I don't want our water to boil off. That's the only thing we need the water to heat it up for. So once that time is up, we'll take them out and let them cool. So they'll be cool by the time you're ready to use them. All right. Yes?
It is not. Yeah, I'm.
Is it what?
Yep.
5 minutes.
Everything is worse than that.
All right.
You should have big air bubbles from the bubbling, and then it's pushing up the, all right. So we have 2 labs that we're doing today that we're starting today. Today's day 1, and Monday will be day 2. These are the last 2 labs for the lab practical. Okay, yes.
One of our tubes broke.
Okay, just grab another one.
So this older thing's gonna be covered.
Grab another one there, and
So these are the things that we'll be covering for the practical exercise.
Can you send me, you've been writing down some notes about what questions we had. Can you send me a picture of it?
all right.
I only did ones.
Huh?
I only did ones.
So
But I
these are
can
the
go
last
ahead
2 labs for the lab practical. So this will be the last that we'll cover, and then it'll be included here, right? Exercise 14 is going to look at motility of microorganisms and how we determine motility, and exercise 15 is going to look at oxygen requirements. Right? So when we are determining motility, there are actually different ways we can do this. So we're actually going to cover both processes. One is to do a wet mount and look at it under dark field. Okay? So we'll do that today. We're working for this, by the way, for the wet mount. You can work as a table. We have 2 organisms, Proteus mirabilis and Staphylococcus aureus. So, just those 2 need to get prepped. And then you guys share the information.
That's sweet. We figured it out.
And then the other is we are going to do the tube method, which is where we're going to inoculate motility test media. In this case, we are going to everyone is going to inoculate a motility test media. So you are going to do one of the 2, and your lab partner the other. For those tables where you don't have a lab partner because you're group of 3, that's fine. The other person, just pick one. I don't care which one. It's more to do the technique, okay? And you'll share and get the information from the other people at the table. All right? So if you have 4 people at the table, you'll have 4 motility test medium. If you have 3 people at the table, you'll have 3 motility test medium. No matter which table, at least 1 Proteus mirabilis and 1 Staphylococcus aureus. You might have more than. Okay? All right. So, for the wet mount procedure, again, as a table, you only need to have, you'll do 2 slides, one for each organism. You're gonna prepare the wet mount just like we have done at the beginning of the term. You'll notice that at your table, there is a transfer pipette. You'll use the transfer pipette to transfer onto a clean slide. And then up here is where we have the cover slips. So, 2 or 3 drops max. You don't want too many drops or it'll just run right off the slide, okay? Then you're gonna put the cover slip on top of it, and you're gonna put it under the microscope at 10x, okay? You will then go ahead and put in your dark field diaphragm, and while the dark field diaphragm is in place, you have to be at maximum, so the rheostat is turned up to maximum, highest light possible. So rheostat all the way up, iris diaphragm all the way open. Okay, you want the most light as possible coming through. Both of these organisms, you will see movement. So if you see no movement at all, okay, that's a problem. You're probably not looking at the organism. Just call me over and I'll make sure you're in the right field of frame of view. But keep in mind that we're the same thing throughout the entire term. It's all the way up, quarter turn down. Okay? Then you're going to make a determination and note whether you see what we call true motility, which means the organism is actually modal, or whether you have Brownian motion. Brownian motion is not motility. Brownian motion is due to the fact that the organism is small
10, 9,
and molecules are there bumping around, moving around. The organism is present. And it can bump into each other. So you might see some wiggling, jiggling type of thing for Brownian motion. Or you might see currents. You have a liquid underneath that microscope, you have light, there is going to be a current as the water heats up, okay? Brownian motion is non-modal. True motility is where you see individual cells moving in different directions, doing different things. They're not moving as groups in a current, and they're not just jiggling. They're actually, as you watch them, they're moving. When you're looking at these under the microscope, number one, the highest we'll go is 20x. I don't know what this says, I don't really care. You don't need to go higher than 20x. You're not putting oil on your cover slip to go looking, okay? It is, you look at it at 10, bring it up to 20. If you're calling me over to take a look at it, make sure you're seeing what you're supposed to see, which is perfectly fine. That's where I want to see it, is at 20x. At 20x, Proteus mirabilis is a rod. Staphylococcus aureus, you already know, is a toxide. And you can actually tell the difference. One looks like a rod. Even though they're very small, they will look like a rod shape for Proteus, and the other one will look like twinkling stars, right? So, the question then becomes whether you making that determination whether it's true motility or Brownian motion. If I get called over to your table to take a look to make sure you have it in focus, that's all I will do. Thank you. You can turn on, once you have those off, you can turn off the water bath and unplug the heat block. What I will not do is tell you if your guess is correct. Okay? So I'm not going to tell you whether you're right or wrong as to whether it is true motility or Brownian motion, because that's the whole point of the tube test. Okay? And you won't get the tube test results until Monday. I know, I'm going to have you get a cliffhanger until Monday. All right? You know that. I knew that. Anyways, the tube test is pretty easy. Again, everyone's going to do one. And that the motility test medium, it's abbreviated MTM. It's with TTC. Okay, and by the way, you might have noticed in the, I've got a location for 171. It is one of the tests we'll do for the unknown. So if you go to page 171, you'll see a lot more detail in regards to the purpose, the description of the test, etc. So I like to reference that. TTC, okay, TTC, I will never ask. I will never ask you what that stands for. And doggone it, at least once a term, I have somebody who wants to try to figure it out on a test that they think I'm asking for. I will never ask you what TTC stands for. TTC stands for 2,3,5 triphenyltetrazolium chloride. Okay? I will never ask you that. So don't try to memorize it. What I will ask you though is what it breaks down to. So TTC is metabolized by the microorganism when it grows and it produces formazine. That I will likely really ask for. Okay? But don't try to sit there and memorize what TTC stands for. Formazine.
What is it again?
Formazine. Okay? It's actually in that description too. TTC says viable bacteria reduce TTC to form a red precipitate called formazine. Okay? It's just going to help us be able to see where the organism is in that media. Okay? All right. Now, how are we going to inoculate this media? The inoculation technique that we're going to do is what we call a straight stab. So, that means you're going to use your inoculating needle in this case. You're going to use your inoculating needle. I know that the cultures are coming from a broth, but that's okay. You're going to use the inoculating needle, and then you're going to straight stab, means you're going to then straight stab down, almost to the bottom, and pull up, hopefully on the same pathway. Okay? So people who's got a little more of a handshake, don't stress. I mean, do the best you can, but you want to go straight down and pull straight back up. That's it. It's inoculated. Okay? As far as the label is concerned, 5 bits of information. So it'll be the exercise 14. Today's date is the 1st. We have, this is MTM. That's as far as I will ever go if I ask for the media name. Motility test medium. I will never ask for the TTC. Don't add the TCC. I'm okay with that. Motility test medium. Okay? And then, of course, you'll have your initials because you're each doing one of these. And what else, guys? There's 537 degrees Celsius. And then, of course, the organism. And that'll depend upon which one you actually assign to yourself. Okay? That's it. You'll go on to the rack up here. There is a rack, white rack that is sitting by each of the originals. Put it in the white rack sitting next to the same media. Because these actually come out a little later than the FTM. The FTMs come out sooner. Okay, that's why we gotta keep the rack separate and the media separate. All right. Any questions in regards to this? Benefit of motility, the wet mount, doing the dark field wet mount, is what we get immediate results. So that's the pro. The con is both, whether they're modal or not, that you will see movement. So you have to be able to differentiate between true motility and Brownian motion. Okay? The tube method with the motility test medium, pro is it's easier to read. Con is you gotta wait for incubation. Okay? All right. Questions on this one? Okay. These slides, when you're done with the wet mounts, cover slip and all in the bleach bucket. Okay? Don't clean those. The whole slide in the bleach bucket. All right, so exercise 15. This is we're going to be looking at oxygen requirements. So, which is what we talked about in lecture today. Okay, so we're following it through with the experimentation. Lined up just perfectly. Doesn't always happen that way. So we're going to use, we have at your table, you have 3 organisms, Escherichia coli, Clostridium sporogenes, and Pseudomonas aeruginosa. We're going to inoculate these 3 organisms into their own tube of fluid thioglycolate medium. Okay? Again, this one is one we will do for the unknown, so if you look at page 172, it also shows this media in a lot of good detail, all in one, wrapped up in one page, right? And so this media, one of the requirements of this media is that you have to boil it first and boil it for 5 minutes and let it cool. Okay? Why are you boiling it? When you boil this, when you boil liquid, you're forcing out oxygen. Okay? So we boil it to force out the oxygen. I'll ask that question. Oh, I might ask that question.
No, you said you go.
You boil it
To force out
and
oxygen.
Add oxygen?
why would you, the question would be, why would you boil it? To remove oxygen. Okay? Don't tell me. Every turn, even though I say don't tell me, somebody will say, oh, because you want to sterilize it. It's already sterile medium. It doesn't have anything growing in it. So you boiling it had nothing to do with removing contaminants, sterilizing, or anything like that. It's already sterile. It was sterile before you put it in that water bath. You're only putting it in that water bath to force out oxygen. Right before we start using the test, we want to force out all the oxygen. Now, are we cooling it? So when we inoculate, we don't kill the organism, right? We're cooling it so that we don't kill the organism before we inoculate. So today, before you inoculate, make sure you can comfortably hold it in your hand. If you can, that's cool enough. Okay. Now, this media, it does have the in it that I talked about today. It has that glucose in it as source for energy. So fermentation of that glucose. It has cysteine and sodium thioglycolate. Those are what's going to reduce that oxygen. So that sodium thioglycolate reduces the oxygen. Why it's part of the thioglycolate is part of the name. Okay. And when you look at this media, I'm going to turn on down the lights for just a moment. When you look at this media, when you guys pulled this media, you might have noticed that there is a red at the top and yellow at the bottom. There is an oxygen indicator in this media called resazarine. Resazarine turns red in the presence of oxygen. And therefore, we can tell where the oxygen is present in this tube. If it's red, then oxygen is present. If it's not red, then oxygen is not present. So we can see in this, the anaerobic zone is here, the aerobic zone is here at the top. Right? The anaerobic is at the bottom, aerobic is at the top. Can you spell resazarine? Oh, it's in the book. It's in the book. Okay?
Hold on. What is
It's spelled much like literally when you sound it out.
Yeah, where is it?
Okay.
Aerobic. The bottom one is anaerobic.
All right. Now, for inoculation of this one, you do have the 3 cultures. So you have 3 tubes, 1 tube for each culture. Okay? So if you have 4 people at the table, 1 person's just not going to get to do it. But the technique for inoculation is the straight stab. Okay? So everyone got a chance to do it with the motility test medium. It's not any different. The only difference is not the technique, but why. In this case, we do a straight stab, and you can use a loop. If the culture is coming from a broth, this time you can use the loop. Because this time, fluid thioglycolate is a liquid, not a semi-solid, right? If I turn this to the side, we've got liquid, semi-solid. This one you're straight stabbing with the needle because we want a line down the center. This one we're straight stabbing because we just spent 5 minutes boiling out the oxygen. If you do a dip and swirl, you're going to introduce oxygen, right? So we straight stab solely so that we don't introduce extra oxygen. Okay? Once you straight stab, you're going to then turn around and do a gentle roll. Okay, just go like this, gentle roll to help mix that specimen or inoculum in the tube. This is going to be incubated at 37. You're going to put it on the white rack that's sitting next to these tubes, right? And the label is, in this case, it's 15. This is table-based. So we're working at the table on this one. So FTM media, and we put the table number. It's still 37 degrees Celsius, and the organism. Okay, that's That's it. Okay, that's the extent of this one. So things I could ask, right? What is the name of the media? What is the dye in the media? What does the dye detect? Right? I could be asking, how do you inoculate this? And I'll even say, don't give me aseptic technique, because that's a given. We're doing aseptic technique always when we inoculate. But this technique specifically is the straight stab. Okay? I could ask you, why we might be doing the straight stab.
Because
Right?
we
You
do
don't
not
want
want to
to introduce
introduce
oxygen.
extra oxygen.
I could ask you why we boil, why we cool.
We want
Okay?
to take
All of
oxygen
those
out.
things are all questions that could be asked. Okay? All right, so this is gonna take, now, those are probably already cool, so this is gonna take, all of about 3 or 4 minutes. Done. Motility and test medium, 3 or 4 minutes to inoculate. The longest thing it's gonna take you is probably looking at the wet mount. You guys can go ahead and start. I'm just gonna remind, wet mount slides go into the bleach bucket. The cultures at the table, once you're done with all the cultures at the table, you remove the label as best you can, and put it on the kill cart. You've already, if you haven't already, turn off the water bath, the hot plate, and unplug it. Other than that, the transfer pipettes, you can temporarily put them into the bleach bucket, but before you leave for the day, make sure you get those, the transfer pipettes, into the biohazard.